Greetings, glory and honor to our Lord and Savior, Jesus Christ. Welcome to the Evidence and Reasons for the Christian Faith video channel. I'm Salvador Cordova, and today we are joined by Dr. Change Laura Tan. She is a professor of molecular and cell biology. She used to not believe in creation. She was born in, correct me if I get any of the details wrong, she was born in China, in Red China, and where they taught evolution. She came to the United States, studied at an Ivy League college in, University of Pennsylvania, and she did her postdoctoral work at Harvard, and which is amazing. She encountered a Nobel Prize winner by the name of George Smith, and George Smith said to Dr. Tan that she should teach molecular biology. So Dr. Tan's background before that was in physical organic chemistry, and then she did biology and then specialized in molecular biology. She'd done
not specialized initially. Actually, I I knew nothing about molecular biology before I was asked to teach molecular biology. So I had to self-teach before I teach my students.
And so, and how long have you been teaching? You taught at University of Missouri, and now at at another university. And I actually
20
passed
years.
20 years. Okay. And I'll share this. Let me advertise your book. It is my favorite. It is my favorite book that I would absolutely recommend to serious students of intelligent design. And the book is Stairway to Life. And if you hear me typing,
yes,
crazily, all right, here it is. And I need to get an autograph from. I have to bring my copy the next time I see you and Rob.
right. As you, my
So I can get.
is beautiful.
So here is the best book. If people come up to me and say, Sal, what would you, if you had one book to recommend on intelligent design, what would it be? I said, The Stairway to Life, An Origin of Life Reality Check by Change Laura Tan and Rob Stadler. Rob's, I talked about Dr. Tan's background. Dr. Stadler has a very high H index. He's a biomedical engineer who got his PhD in a joint program between Harvard and MIT. And I said, well, we've got two highly educated individuals and successful scientists writing this book, and both are intelligent design proponents and creationists on top of that. My understanding is that once you started teaching molecular biology, you began to have a different viewpoint about things. Is that correct?
That's right. Yeah, I was amazed that actually what I thought were true is questionable.
Yes. And I really do hope sometime we can talk about some of your work, both on eukaryotic, prokaryote, eukaryote transition, the Stairway to Life. And then there is one project that you had worked on, the 1,000 Genomes Project, analyzing the work of other scientists. And my jaw dropped. If I may mention this, because people on the internet had been asking me about this, and I may have said something incorrect, or maybe it was correct. And so before we get into the bulk of today's topic, maybe I can clear this up. So let me see if I can. It was the 1,000 Genomes Project. And I'm having a hard time finding it on the web here. Let me see. Let me search under a different term. Here it is. It says here, okay, let me share my screen and then we'll get to today's topic. It says here, oh wait, there it is. That's a little bit small. So I'll increase the, not a single protein is conserved across all genomes. Now, that wasn't something you said. It was in this article by Microbiology Research. And the article's name is, hang on, my computer's a little slow here. Genome Update, the 1000th Genome, a Cautionary Tale. So that's a quote from this peer-reviewed paper. And you'd been able to contact some of the authors. So the question I had, I mean, this is amazing. Not a single protein is conserved across all genomes. And then I remember asking you, what about the polymerase? And you said, well, the polymerase is made of subunits. I said, well, surely a polymerase subunit was conserved across all genomes. What was your take on that, or has that issue been resolved?
There is regions of the polymerase are conserved, right? So also depending on what your definition of conservation is, your threshold of calling something conserved versus non-conserved. Depending on that, right? Okay. So you can call something conserved or not conserved.
Okay, so thank you for setting that straight. That would be something worth exploring later on. And to the extent I have misstated the facts, apologies to all the viewers. Someone was asking. And I may have misstated and said, well, there's not even polymerases are conserved. So I stand corrected there. So thank you. So now how can I be of service? I received an email from you. And we talked backstage a little bit about it, but perhaps you could share it with our viewers, what you wanted to ask me.
Well, I'm really trying to understand what evolution is by any standard, how the ideas are developed. For example, we have Mendel and Darwin almost at the same time working on speciation, how different species come about. And then later on, we have Mendel's idea and Darwin's idea initially seemed contradicted with each other, and later on they were combined and say, oh, actually they are compatible with each other. That's one thing. And the main thing is, what is the key issue? Really, what is the root of the division between, for example, evolutionists and creationists? What both groups are trying to strive to understand about life.
And could you show the book that you're presently reading now?
I have this book by Dr. Ernest Mayr, The Growth of a Biological Thought, Diversity, Evolution, and Inheritance. So I'm trying to read some of the classical books on evolution, trying to understand the history of evolution, the variation of thoughts, and the progress of ideas.
So this is just for the benefit of the readers, because there are also I have plenty of detractors. I probably have a ratio of 10 haters for every one person that likes what I have to say. And I do want to try to set some things straight. Now, you were teaching molecular biology. I presume you're curious how a prokaryote could evolve into a eukaryote. Now, specifically in molecular biology, correct me if I'm wrong, you're looking at genetics, transcription, and translation, basically expanding on the central dogma of molecular biology. And you must have started looking at evolutionary explanations for that. Is that fair?
Yeah, well, initially, of course, I believe that life came from no life, and complicated life evolved from simpler ones. Especially eukaryotes evolved from prokaryotes. When I was preparing my molecular biology lecture, which basically deals with the DNA replication, transcription, and the translation. So I'm looking at how bacteria and archaea do their DNA replication, transcription, the translation, and how eukaryote do the same thing. What molecules are involved. And to my big surprise, the molecules involved are mostly unrelated in sequence. So that made me think, how could that be? Because even though I was not trained as a biologist, I was trained as a chemist. I knew DNA replication, transcription, and the translation were essential for life. So those vital processes, how come, prokaryotes and eukaryotes, especially bacteria and eukaryote, they are so different. They use really the logic behind it is very distinct, right? So you do the same thing, same task, but the implementation, the genes involved are mostly unrelated. So that made me think, how come that, prokaryotes could evolve into eukaryote? Or maybe even if we started with eukaryotes, how could eukaryote devolve or simplified to prokaryotes? You cannot go either way, right? Because before you reach to the other side, the organism would die. And because each of them needs their unique essential components, genes, many genes involved, required to replicate the genome, recognize the gene, transcribe their gene, and translate those genes into proteins. So that was my starting point for thinking the issue of evolution, right?
It's a very good starting point, because when people will talk about, the fossil record or chimp human ancestry, I say, look, let's grant that. What about the most important transition? And I'm going to quote here. Let's see if I can find this. Echoing that, and I'm just going to share this with the viewers. Sharing my screen. It's a slow process to share screens on StreamYard. Anyway, this is from an article about the title of the article is The Long Winding Road to Eukaryotic Cells. The Long Winding Road to Eukaryotic Cells. And one of the researchers said, and the article points this out, Leger agrees that our understanding of eukaryogenesis is likely to advance with baby steps. Part of the nature of these deep evolutionary questions is that we will never know. We will never have a clear proof of some of the hypotheses we are trying to develop, but we can keep refining our ideas. So I just
yes. So sometimes we say we can never know, and then other times we say, "Oh, we're sure, definitely, right? We're here, so we're here. We're eukaryotes. Therefore, evolution must have worked because we're here."
Right, which isn't very good reasoning. I mean,
Is it?
if you say it worked, well, something worked. I mean, if you invoke an intelligent designer and miracles, it's like evolution worked because of miracles. And it's like, okay. But what they're really saying is it happened without an exceptional process. And
It
then,
all happens naturally. That's the point.
right. And there's a way to actually frame it, I'll say, okay, I'm just speaking for myself here. People say, do you mean supernatural cell? I'm like, look, scientifically, we can determine if something is operating within normal expectations. We have distributions like the binomial and normal and all sorts of other distributions. If events have to happen that are at the tails of the distribution, it's kind of hard to assert that this is a normative process. And that's what I'm just going to say. We don't have to get full-blown metaphysical and invoke miracles to at least say what is required are events that are at the tails of the distribution. And once you get up to about 20, 30, 40 sigma, it's like, okay, this is like one chance in 10 to the 150 power. When you get even farther out in terms of, how many standard deviations from what you consider normal, it's just super astronomical. And we can, as scientists, say, okay, given what we know about how chemistry and molecules behave and inheritance, etc., we can make a pretty good guess of what events had to happen and maybe how far away from the expected norm, the expected mean of a normal distribution or whatever distribution. You can't just say this happened normally. If it took exceptional events, that's something to consider. And that's why I really liked your book, Stairway to Life. So I presume after you started asking all these questions, you began to be maybe a little more favorable to a miracle.
Well, I'm not that upward looking. I'm not as metaphysical as you are or mathematical as you are. For me, it's all genomes, and what is essential for life. So basically, what is essential or necessary to keep something alive, right? For anything to evolve into anything else, the organism has to be able to stay alive and to reproduce, right? If you cannot be alive and you cannot be reproduce, how can you make something else? Even, better or worse, right? How can you make something else if you are dead and you cannot be alive? You cannot reproduce. That's the only thing for me. I'm not, I cannot think very abstractively or at the high God level, right? For me, is very basic. Okay, stay alive and be able to produce. That's all.
Well, and from a biochemistry textbook, it was nice to see this. McKee and McKee's Biochemistry, the Molecular Basis of Life, one that our colleague Joe DeWeese, by the way, it was through Joe DeWeese that we met, as far as we can tell. It says there, Virchow's principle, all cells come from pre-existing cells. There are probably different ways that this is more properly phrased. But if all cells come from pre-existing cells, then where did the first cell come from? That was the basis of, that was one of the questions that was explored in your book, Stairway to Life. But if I'm getting the chronology correct, if my understanding is you first started not with the origin of life problem, but with the prokaryote, eukaryote, or no one, hardly anyone believes this, eukaryote to prokaryote. But that transition, whatever it was, however it happened, that raised questions in your mind, and that also naturally led to studying the works of Craig Ventnor and then the origin of life. Is that correct? Is that
Yes,
fair?
yes. So for me, is we're giving you all the prokaryotes, right? All of them. Whatever you need, all the prokaryotes, I provide to you. Can you make a eukaryotes from all those prokaryotes? That's my starting point.
Okay, so the reason I'm reaching out, we had to go into the past to everything. I wanted the critics of creationism, the first thing they'll say is, well, Dr. Tan knows nothing about evolutionary biology. And I just wanted to say, look, in her defense, she's taught molecular biology for 20 years. And I've just provided evidence here that even professional evolutionary biologists have said, just as I pointed out, they have said, part of the nature of these deep evolutionary questions is that we will never know. We will never have clear proof of some of the hypotheses. You have it right there. And yet, in the way that you would think these evolutionary propagandists go around, they're saying, this is so proven, it's as certain as gravity. But when you actually talk to professional literature versus the popularizers, you get a different story. And so Dr. Tan's approaching me about, she wants to learn more about evolution. And I think, what would I like to teach her? So there's a verse in Romans, it's in the later chapters, chapter 14 or 15. It says, why don't I quote it? It's a beautiful Bible verse. Hang on.
Everything Google, right?
I'm using Google.
Oh,
Romans.
you are using ChatGPT.
No, 15:14. This is one of my favorite verses. It says, this is what we're going to do today. I'm going to show this verse just so everyone can see that I'm quoting the Bible. And I'll just show it. It's actually just one verse. And I appreciate the audience forbearance as I'm very slow at getting screenshots up. But it says in Romans 15:14.
15:14.
And there's a variety of verses, but I'll start with the New International Version. The Apostle Paul writes, I myself am convinced, my brothers and sisters, that you yourselves are full of goodness, filled with knowledge, and competent to instruct one another. Another one says, filled with all knowledge and able to instruct one another. So Dr. Tan has instructed me in molecular biology, in prokaryote, eukaryote, and then the Stairway to Life. She's one of my favorite instructors.
And so I feel me about evolution, about the fishes.
So this is right. So this is just from the Bible. This is normal. We are able to instruct one another. And so what I would say to you is what an evolutionary biologist said. The nature of these deep evolutionary questions is that we will never know. And we will never have a clear proof of some of the hypotheses. That's my starting point. And honestly, as someone who's been interested in science, who studied physics, studied chemistry, biology, engineering, I don't think I would ever want to enter, or math. I also studied mathematics. I don't think I would be that interested in entering a field that said, we'll never know, we'll never have clear proof. Okay. If I were, obviously they're faith statements. I mean, I go to church. By the way, this background, this used to be a church before it got converted into a bar. But there are things we will believe that we may not have clear, immediate proof. But as far as if I'm going to invest time in a scientific theory, especially a theory that says this is proven beyond all shadow of a doubt, and then I find out statements like this, I'm like, I don't think I'm quite that interested. I mean, since you're a chemist, I mean, we had the periodic table. That's so easy to establish that this is a legitimate way to look at how the world is structured. So that is my opening statement as far as studying evolutionary biology. You're going to hear many opinions, and there's not a unified opinion. Someone may represent that their viewpoint is the majority view, or one could get the impression it's the majority view. It may have been a majority view at one time, and then it no longer is. That's one of the problems. It's hard to pin down what evolutionary theory actually says. It's starting to fracture, actually. It's becoming really evident because I'm seeing there's this paper. Let's see. It's called What is Wrong with Evolutionary Biology? There's a paper, peer-reviewed paper, and I'm going to show the title. And actually, I was really disappointed in the paper because they didn't even touch on the difficulty of the prokaryote-eukaryote transition, or the origin of major protein families, or major features of cells. How about the formation of the membrane-bound nucleus, or the spliceosome? Didn't even touch on that. But there is a paper here that, hang on, I'm sorry. I'm going to share my screen. And then we'll get to the discussion about evolutionary fitness. But here it is. Here's the paper. What is wrong with evolutionary biology? And it's written by an evolutionary biologist, by the way, John J. Welch. And there have been periodic claims that evolutionary biology needs urgent reform. And this article tries to account for the volume and persistence of this discontent. To achieve, why don't I read the whole abstract?
Go ahead.
There have been periodic claims that evolutionary biology needs urgent reforms, and this article tries to account for the volume and persistence of this discontent. It is argued that a few inescapable properties of the field make it prone to criticism of predictable kinds, whether or not the criticisms have any merit. For example, the variety of living things and the complexity of evolution make it easy to generate data that seems revolutionary, e.g. exceptions to well-established generalizations or neglected factors in evolution, and lead to disappointment with the existing explanatory frameworks, with their high levels of abstraction and limited predictive power. It is then argued that special discontent stems from misunderstandings and dislike of one well-known but atypical research program, the study of adaptive function, in the traditional of behavioral ecology. To achieve its goals, this research needs distinct tools, often including imaginary agency and a partial description of the evolutionary process. This invites mistaken charges of narrowness and oversimplification, which come not least from researchers in other subfields, and these chime with anxieties about human agency and overall purpose. The articles discuss several ways in which calls to reform evolutionary biology actively hinder progress in the field. And he quotes one biologist. One biologist, evolutionary biologist, said, "I've been wondering why so many evolutionary biologists are unimpressed by my idea and think the problem goes back to Newton." And so John J. Welch says this: "A dispiriting thing about working in evolutionary biology is the steady stream of claims that the field needs urgent reforms. Those critiques are too numerous to cite, but representatives include Waddington, Moorhead and Claflin, Hoe and Saunders, Gould, Pigliucci and Muller and Lalland et al."
Actually, I have been studying the extended evolutionary synthesis for the past few weeks and the modern synthesis. That's why I bought the book, I showed you earlier. So I can read that. I do also have a copy of the modern synthesis by Huxley. So trying to see really what is going on, what is behind the theme.
If I had to summarize everything, it goes back to that comment. We will never know. We will never have clear proof, but we can keep refining our ideas, which means we'll never know, we'll never have clear proof, but we can keep finding ways to argue with each other as evolutionary biologists. So
I think it's okay. We don't know things.
So one of the articles,
we are limited. So it is fine that many things we do not know. We admitted that we are limited and we don't know things, and that's how we trying to do scientific research. So we keep on study, we learn new things, and find answers. That's fine.
Well, I will tell you this: in physics, in the discipline of physics, we have Heisenberg uncertainty principle. It actually tells us certain limits of what we can actually know. And so a lot of physicists are like, okay, well, we're not going to go beyond that. We're not going to try to predict the position of an electron simultaneously its momentum. And it ended up being a law of physics. Okay, the Heisenberg uncertainty. So
very strange. I learned that
what
too.
Is troubling, what is troubling to me is they just keep going on and throwing up these theories. It gets quoted by the popular press, and then it's not really highlighted that they'll never know whether it's true or not. It's unprovable. So I'm just sharing that with you. Some of the things you may be reading in evolutionary theory are just asserted, and then they're quoted by other evolutionary biologists as authoritative. And yet there could be no proof in the end, but the literature is just rich. It's kind of like studying theology, or mythology. It's just rich in terms of the amount of output. And further, it is being reinforced by peer review, official peer-reviewed publications. In fact, when I read about the long winding road to eukaryotes, these are paid researchers, and I'm presuming they're publishing. It is nice to know that they say we may never be able to prove or know. I hope they put that really in every peer-reviewed paper that they write when they really can't ever prove or know. So when you read all these books, I'm just wanting to say that's my caution to you. You have kind of an instinct, as you demonstrated when you were looking at teaching molecular biology and raising these questions. You might, after reading all of that big book by Meyer, come to the conclusion that there are probably a lot of things that will never be proven or known. Okay, it's still worth it to read it. I have kind of my limit of how much of that I could personally take, partly because there's so many other things I could study, like your work. I have read Stairway to Life through once cover to cover. Actually, yeah, at least once cover to cover. I'd love to read it at least another time, and then also go through your molecular biology course. I say that sincerely. There are areas of physics that I'm reviewing. And so I'm just saying I have my limit of how much I can take of reading stuff that even evolutionary biologists will, on the side, in an honest moment, say, we can't prove this and we'll never know. So that being said, you had asked about learning about Fisher and fitness. Okay, historians, the first problem is historians of Fisher's work don't necessarily agree on the significance of some of his claims. So specifically with regards to Fisher, there's this famous theorem, Fisher's fundamental theorem of natural selection. And it involves studying, you have to be able to see the meaning of it, one has to understand the population genetic definition of fitness. So I'll preface this. Why don't I give you if I may encourage you, the reading is really, really, really thick. There are 2 papers by Lewontin. One is a little easier to read. The other one's just absolutely horrible. But we can read through a few parts. So let me see if I can find, if you'll give me a moment. If you wanted to say something while I'm desperately searching.
Maybe you should give us the links to those 2 articles.
Okay, that's a great. Yes. So
yeah, for me
I have the
is to know what the author really says. We need to go back to what they really says, not by just hearing somebody else talk about what they said.
I'm so glad you said that. People have been criticizing me. It's like, your students, are you going to be honest with them and link to all the papers? Well, I have, as one of my students today, a professor of molecular biology, a Harvard postdoctoral fellow. And you gave the answer that I'm so delighted to hear. You want to read it and study it for yourself. So for all my critics that say I'm just quote mining this and I'm trying to deceive, you've got a professor of molecular biology, a Harvard postdoc right here saying she wants the links and doesn't want to just hear my commentary on it, which I'm thrilled to hear. I don't take offense. I'm thrilled when one of my students would actually say that to me, because then I won't be accused of quote mining or trying to hide all the details that may be contrary to my viewpoint. I'm going to lay it out there. So I will provide you a link and I will share with you. I actually did this on the internet. And it is such a pain to show links, to share my screen on StreamYard. Hang on. I will share this with you, but for the benefit of the onlookers, I'll also put this in the video description below. But let me give some selected quotes, which I think are representative of the themes. You can decide for yourself how accurately or how better to say what I said. So I have said, what is the fundamental problem with evolutionary biology? So Weiser and Lenski wrote, they said the concept of fitness is central to evolutionary biology. I got some pushback from evolutionary biologists or propagandists. They said, no, that's not. No, it's not. I'm like, well, look, Lenski said the concept of fitness is central to evolutionary biology.
How do you define, or they define fitness here?
Okay. And then I'll show how I think they defined it. They actually have a journal where they, in their journal article, they describe how they calculate fitness. And it only really works, I think, for bacterial populations. It's hard to do for something that has a long generation time like humans. And I'll get back to that question. If I forget, remind me. Now, so they said that the concept, so Weiser and Lenski said the concept of fitness is central to evolutionary biology. But then I quoted Ariu and Lewontin. Lewontin is, you'll probably read about Lewontin in your book by Meyer. They were colleagues, I think. They both taught at Harvard. It wouldn't surprise me if their time there, they overlapped. They may have been on the same Harvard campus while Ernst Mayr was alive. He lived to be 100 years old. Now, Lewontin wrote, no concept in evolutionary biology has been more confusing. No concept in evolutionary biology has been more confusing and has produced such a rich philosophical literature as that of fitness. And I'll give you the link to that paper and you can read it. Now that's a really painful paper. One that is less difficult, written by just Lewontin himself, the solo author. He wrote this a year earlier before the larger article. He said, the problem is that it is not entirely clear what fitness is.
So we don't know what fitness is, and we know that all fitness evolved by natural selection.
Okay, that is my conclusion. Zachary Hancock and other evolutionary biologists pounced on me and said I'm quote mining or whatever. I'm going to let, oh, by the way, Dr. Tan, you're my star student. Just want to let you know.
Thank you.
I'll let my star student investigate and make her own opinion. And he says here, Darwin's sense of fit has been completely bypassed. He points out the way population geneticists today define fitness is not the way Darwin meant it to be. And I'll give you the link to that paper. Andreas Wagner, after reading Lewontin's work, he wrote, fitness is difficult to define properly and nearly impossible to measure rigorously. An unassailable measurement of any organism's fitness does in practice not exist. And I'll give you a link to that, and you as my star student can review these papers and tell me what you think. But this is my conclusion. So I say, so the central concept in evolutionary biology is the most confusing. It is not entirely clear what it is. It's difficult to define properly, nearly impossible to measure rigorously, and an unassailable measurement of it does in practice not exist. So I'm just stitching together what all those quotes had come from those papers. And I say, okay, as me as a student of physics, contrast this to the four fundamental quantities that are measured in physics, from which pretty much all the other physical units like force, pressure, velocity, acceleration, electric current, voltage, resistance, etc. are constructed from. And the four fundamental units are mass, charge, length, and time. Mass can be measured in grams, charge in coulombs or electron charge, length can be measured in meters, and time in seconds. But evolutionary fitness, that's why we have titles like this by Lenski in peer-reviewed literature. This is a shorthand title, Genomes Decay Despite Sustained Fitness Gains. That's why.
Still, so in physics, all right, when you keep on going down to to the tiny particles, the subatomic particles, then you have the principle of uncertainty. You goes back to the, well, we don't know what is light, for example. Sometimes it behaves like particles, sometimes it behaves like a wave, and we cannot be certain about its speed of movement and its location at the same time. So you also have some uncertainty physically in the physical world. So why
What's the difference?
what's the difference? We have some
Right.
uncertainties in evolutionary theory, right?
Okay. So when we've done experimental work, we always say, okay, we have, we have a certain number of significant digits. The rest is kind of like, well, there's uncertainty there. And so we often will state when you make a measurement, it's plus or minus this range. And that's how a lot of things. So if we measure a length, we'll report it as like 5 meters plus or minus this. With the wavelength of light, we'll say it's this number of nanometers for this particular frequency or whatever. Any, like if we have, there are a variety of times we will put plus or minus. We have we have at least a good attempt to define the uncertainty.
Okay.
And so what you posed as my star student was a very pointed question, and that's my best answer. So the fundamental units of physics are mass, charge, length, and time. We can also put the uncertainty. And as how this came about also is I have friends, physicists who've worked at the National Institutes of Standards, NIST, NIST, and they obsess about how we can, define the mass, the rest mass of an electron, so to speak, or how do we even define what a second is? And then they will actually define it in terms of some sort of atomic thing and say, okay, this is the standard we will use by which to judge all the other clocks, or we'll use this as the measurement for all the other lengths. So we're going to define the meter like this. And they have they have professional physicists spending their lives trying to figure out the best standard of measure. You're going to find out that fitness has none of those characteristics of precision and accuracy. It is a very disturbing thing, very offensive to my sensibilities, having been in a community where people are spending their lifetime. I mean, in a community where I met people who spent their lifetime building more accurate clocks, like these cesium beam atomic clocks that can go to several decimal places in precision. It's amazing. And then I go and I ask questions about fitness. So I'll give you an example. I really want to ask one of our colleagues. We have colleagues who are medical doctors, like Howard Glicksman. You know Howard Glicksman. He wrote that book. I'm embarrassed.
Your designed book. Your designed body.
Your designed body, along with Steve Loffman. So Howard Glicksman's a medical doctor. I'd like to ask him, Dr. Glicksman. In fact, I'm going to bring him on my show sometime. I'll say, Dr. Glicksman, if you're doing a medical exam of someone and you found out he had sickle cell anemia, or he had a sickle cell trait? There are two varieties. One of them's homozygous, and the expression is homozygous or heterozygous. Homozygous is really bad. Heterozygous is okay. Now I'll say, Dr. Glicksman, would you say if you had an individual that had a sickle cell trait, like, would you consider that a fit trait? Would that raise concerns for you in terms of that individual's physical fitness? Or how about Tay-Sachs disease? Or how about high cholesterol? Or a tendency toward diabetes? And I okay, if it were me, I'd say, if I had, not that I've ever been a doctor, medical doctor, although mom really wanted me to be one, I'd tell my patients, sir, I do have some concerns. You've inherited these traits. It can adversely affect your fitness. So, what did the evolutionary biologist have to say about fitness? When I was in a debate with an evolutionary biologist, he cited sickle cell anemia as proof that fitness can increase.
Yeah, I know. That's silly, right?
Survival, and there is this title of
Definition, condition.
Okay.
Context.
It's the context. Now, in physics, if we're measuring mass, okay, as long as just to be technical for those who are really persnickety about details, okay, this is not for your sake as much as it is for my physics colleagues. If we're both in the same inertial frame, the rest mass of an electron is always going to be the rest mass of the electron. You're not going to have, it doesn't matter what environment you are in, provided you're in the same inertial rest frame. That's not going to change. It is bothersome to me that in one environment, the sickle cell trait is considered fit, the heterozygous version anyway. And then in most of the rest of the world, it would be unfit. Where is the objective quality? You can try to objectively qualify it, by saying, okay, in this environment, it's fit, in this one, it's not. But when you actually look at a lot of evolutionary literature, when Darwin said survival of the fittest, is there any qualification when most people say that? It's like, well, the fittest in one environment could make it totally unfit in another. So at a presentation which you were at, before I get to that, if you could, I'll try to hold that thought about the presentation. I'll get to that. What I really wanted to show, there is this funny book, and I'm gonna share the title here. It's Survival of the Sickest. It's a New York Times bestseller. And it says, a medical maverick discovers why we need disease. And so this long
wow.
Okay, I said sickle cell anemia, Tay-Sachs disease, diabetes, obesity. And he's going on and on about, oh, this is such a genius thing that Darwin discovered,
It's funny, actually. I read some article recently about one of the recent trends of thought on evolution was survival of the luckiest. So I cannot remember the name of the author. He said, not the survival of the fittest, not the survival of the sexist, but the survival of the luckiest. So basically, survival of the fittest is a natural selection. So survival of a sexist is sex selection. And but the two, right, natural selection and sex selection, actually, in his view, is some kind of a contradict with each other. So at the end is you have both. So the end result is a survival of the luckiest. Right. So basically, he's saying random chance is the bottom line of all evolutionary processes.
Okay, I think I found some of the stuff I wanted to share. And so you'd seen these slides before when I gave a talk. So this one's mutator. I'll highlight the red. Genomes decay despite sustained fitness gains. And see if I could find some more. This is not hard to understand why this would happen. So we have a hiker here who has a big backpack. And that makes it really hard to be mobile, but you're equipped for you have versatility to engage all sorts, a very wide spectrum of environments, like environment without water or shelter or food, very big, well equipped. But the problem is it's not comfortable moving around with all that equipment. So this lady is very well equipped. Another lady would just have a little purse here and it would be adequate to move. The problem, so what happens many times is organisms in a population, the organisms that start to disable genes and not have to waste metabolic energy to express them, they will be more reproductively efficient. And according to the common definition of fitness, more reproductively efficient, anything that's more reproductively efficient is more fit. But what ends up happening is they did a study that in Lenski's experiment, there are a variety of experiments, but the general experiment, they had all these lines of bacteria that grew and became more fit in that restricted environment. But when you put it, so this is the increase of fitness in terms of reproductive efficiency. So one way, it's really nice to be able to measure reproductive efficiency with bacteria because you just put it in a culture and you could see how quickly it grows because it multiplies so fast. And you can actually count the individuals. And then like you could take a bacteria and put it in a petri dish, watch it grow, and you have like a stopwatch. There are various methods of how they actually count this. And then you could take that same bacteria, put it in another petri dish and throw antibiotics and see if it inhibits growth. And you'll say, okay, the antibiotic, it's not as fit in that environment. And so that's how you're able to compare. That's one way you can measure fitness. And so they started to have, as the generations progressed, as it's evolving from 0 to 20,000 generations, as they were carrying out the experiment, the fitness increased, meaning the reproductive efficiency. And so one of the papers I linked to is Lenski, how he explains the actual methodology that they do this counting. What they found out, and it was not at all surprising to me, is to become specialized for one environment, you're going to be very unspecialized. You're actually going to lose ability to work in an environments requiring versatility. It's again kind of like something that's very versatile. It's going to be very hard to replicate it because it has so many objects to make. If you dump that, then you're going to be able to move faster, or by way of analogy, reproduce faster. But then when you're confronted with an environment where versatility is needed, it ends up being not so good.
Yeah, and then you freeze to death, right?
Oh my goodness. Okay, so you're from China, you're mainland China. There's a Taiwanese woman. Since we're both Asian, I'll say, we're both from kind of the same region. I'm from the Philippines. She was known as the bikini hiker. She would go around and hike in bikinis and take pictures of herself, put it on social media, and she got a huge following.
Wow.
They knew her as the bikini hiker. And she died of, I'm not trying to make too light of this, but it does seem funny. She died of freezing.
Oh my, wow.
Now, granted, she tried to have a little more clothing than a bikini on when this happened. She, in her defense, she fell off the cliff. But social media turned on her, and they're saying, well, see, that's what happens when you hike in just a bikini. And she did have this big backpack once, but then she would take it off and just pose in front of the camera with just wearing next to nothing except a bikini. So when you said you freeze to death, I'm just like, I thought of that. That's exactly. And there is a picture of her. The viewers interested, just Google bikini hiker. You'll see this woman in a bikini with a big backpack. And then all the maybe not so nice comments about her freezing to death on one of her hikes. So what does this mean? When Darwin said survival of the fittest, when it didn't have any environmental context, he used that to argue that the process of survival of the fittest will work like selective breeding by intelligent human beings. And that slight variations, you can leverage that to make a more and more complex creature. He used the word origin of species, the phrase in Origin Species, chapter 6, organs of extreme perfection, that it would gradually accumulate the process. But what we're finding in the genome sequencing used to cost $1 million times more than what it costs today. So just imagine a house you could buy for $1 today. It used to cost $1 million. I know that seems outrageous, but this is what's happened to the price of genome sequencing. So now, and then with all these other assays, these experimental setups that make it cheap to run experiments to actually maybe test quote unquote fitness, it's becoming evident that survival of the fittest in terms of reproductive efficiency is actually anti-correlated with the rise in complexity. So when you lose genes, you're losing complexity. And so that's one of the most objective metrics of complexity loss. You lose genes, you lose a capability. If you lose an enzyme that helps you metabolize something, yes, according to the evolutionary biologists, you've become more adapted to your environment, but you've lost complexity. So survival of the fittest, actually, the way that evolutionary biologists themselves have defined fitness may mean that you're less on your way to achieving organs of extreme perfection and complication because you've lost genes. And this has been borne out more in peer-reviewed literature. And let me see if I could find some titles just to, oh my goodness, this is so funny. Okay, I'm going to share some titles with you, my star student, and my other students out there. This is evolutionary biology, and we can, since you're a scholar and my star student, I know you're going to read this and we can talk about it, and you could help share with me how maybe I misunderstood something and vice versa, and we could talk to people. One thing I will tell you, evolutionary biologists do not want to try to explain things to us if they know we're creationists.
Well, actually, I am a big fan of Darwin's Origin of Species book. I also recommended to my students to read the book because it was very well written. So it's a good book to read. What is surprising is, how Darwin examined about artificial selection and thinking that he could apply what people learned or he learned from artificial selection and applying that to natural selection. But in reality, artificial selection and natural selection seems to work all demands on different principles. Artificial selection, you can always select whatever you want, right? Whether it's the weakest or the worst, right? So you can, because you can take care of them. When you select what you want artificially, normally actually is selecting those ones that are weak, right? They cannot survive. They cannot, survive in the nature environment. You make them, you populate them in big quantities. So natural selection does not work that way. If really you live things naturally by themselves, indeed, the weak ones would die, right, without any intervention. But with artificial selection, with a human intervention, then you can keep the weak ones. You select the weak ones, not the strong ones.
So I will show you my, and you've seen these slides before, and I'm happy to dialogue with you some more. So I already talked about the confusion of fitness. No concept in evolutionary biology has been more confusing. And then this is Lewontin's other work. The problem is that it is not entirely clear what fitness is. This is an easier read. It's still a little bit hard. And I shared this one in organisms. Fitness is difficult to define properly and nearly impossible to measure rigorously. An unassailable measurement of any organism's fitness does in practice not exist. And from a population genetic textbook, there are frequencies trying to measure the fitness contribution of, say, an individual gene locus. He said, fitness differences between genotypes, which may be responsible for some of the frequency changes, are so extraordinarily small, probably less than 0.01%, that they are impossible to measure. That's from a textbook. Okay, that's and you saw this quote already. Here's one. 2016, evolution by gene loss. And PLOS Genetics, 2012, selection-driven gene loss in bacteria. Oxford Journals, Molecular Biology Evolution, 2020. Gene loss predictably drives evolutionary adaptation. And do I have another one? This is from Nature, February 2005. One evolutionary biologist who rejected Darwinism by the name of Salty, he pointed me to this article. The power of natural selection. Adaptation by natural selection is the centerpiece of biology. Yet evolutionary biologists may be deluding themselves if they think they have a good handle on the typical strength of selection in nature. By the way, I just for fun, I went to
Actually, the question is why, right, with all these realization of the problems, why we buy into the idea still, everything evolve and the things are getting better and better. Whatever survived are better than those who or what did not survive.
Well, they may be successful in that one given environment. So just imagine someone's trying to climb a mountain or hike a trail. The bikini hiker will get very far ahead of everyone else until there's adversity. So
on the one hand, we want to understand or explain, how life have improved from very simple, unsophisticated to more and more advanced, more and more complicated, more and more able. On the other hand, they will say, well, actually, no, we get better by shedding genes, by losing what we gained by hard work or long time of waiting period.
And see, this is why you're my star student. You echoed exactly the next paper I wanted to share.
Wow, wow.
You know what? I'll say, see, because
People
you're my
may suspect that you have shown me previously, so I can question you, right?
yes. See, your great minds think alike. So, what you have articulated, the number one evolutionary biologist on the planet, according to my estimation, and I think a lot of others, Eugene Koonin has published 1,023 publications.
I
At least,
admire him a lot. He thinks deeply a lot, right? He thinks very deeply, but sometimes I feel like he's so close to the real
right,
true answer, but then he somehow, I think maybe it's limited by his worldview, so he just cannot,
right.
make that extra. Seems like a small step, but he's so close. So I have read a lot of his articles, and a
Oh, okay.
lot of my ideas actually came from his researches, his ideas, his articles.
So maybe that's where you got this, but I'll still credit you. Great minds think alike. So this was 2013, Genome Reduction as the dominant mode of evolution. Quantitatively, the evolution of genomes appears to be dominated by reduction and simplification, punctuated by episodes of complexification.
Hmm.
And I inserted the
Right,
word unexplained.
right.
Um, and and so what I was describing, there are two modes. Natural selection can actually force or induce or favor gene loss. So that's genome reduction by natural selection. Conversely, sometimes natural selection is too weak. A defective trait may end up in the population. That trait spreads because natural selection doesn't prevent it from spreading. It then fixes into the population. So you can have gene loss because of natural selection, and you can have gene loss despite natural selection. What counts is experimental observation. Whatever cost it, however we want to frame it, in the era of cheap genome sequencing, independent of how we define fitness, the experimental objective facts is we could see we've definitely lost genes. That's the most, to me, that's one of the most objective measures of loss of complexity. But then we can, there's some other things where you might have a gene and some other things happen, but we're not able to trace, but there's loss of capability somehow. For example, like people have, there is persistent medical conditions like nearsightedness and so many other things that there is juvenile diabetes, both in dogs and in human beings. Why did that persist? Why does that trait, why hasn't it been eliminated completely out of the population? And some serious geneticists, evolutionary biologists, have been very concerned about the health and progress of the human genome. They're very concerned we're going to be losing. One geneticist, Kondrashov, he said, why haven't we died 100 times over? He tried to offer his solution to that question, which is, I think, since been refuted because we're actually able to sequence genomes now, and we're very concerned about the rise of heritable diseases. And there's an expectation, as far as I could tell, that the rise of heritable diseases will continue to grow within the human population.
So are you saying that in reality, experimentally, we see that mutations, diseases are bad because they, cause pains, troubles, and but on the other hand, we need mutations to accumulate or create new genes, new phenotypes, so making organism better and better. So how do we reconcile our experimental observations and our own experiences to our theories about the history of life, how life came to be, how life have changed?
Well, I think that would be a nice ongoing research project, and you probably know where I lean on
That's
this.
always a good answer, right? Where is
Right.
a research project? We don't know all the answers.
And anyone wanting to grant me a research grant, Dr. Tan and I would be very happy to accept a very nice hefty one. But if I may, can I read the whole abstract of genome reduction? People have been saying I've been quote mining this. And I said, okay, should I read they said, are you going to quote mine that to your students? I said, okay, how about I read the abstract? Would that be quote mining if I read the abstract? They didn't want to answer, it's like, okay, so I'd have to read the whole paper and then you'll be satisfied.
Right, maybe
So
you need
asked
to read the whole
I said,
paper.
Yeah. So I said, okay, would you feel the abstract is a fair representation of the author's view in the paper? Of course, they're not going to say no.
Do the abstract.
I'm sorry?
If it is a good abstract, it should reflect what the whole paper says.
Okay. And we can actually, it might be worth it to go through this once, and then I could talk about a few things where I think the wording was kind of not so clear. And sometimes, I'm just going to say this. Sometimes when an author is going to say something they know they could get punished on, they'll deliberately use wording that's not easy to understand because they don't want to draw attention to the fact they're saying something that's going to be very unpopular.
Okay.
I think Lewontin's paper on the confusion of fitness qualifies. It was deliberately written so no one except the most determined would actually understand it, because he knew I think he would have gotten a lot of pushback if people really understood carefully what he said. Anyway, Kunin, Wolf and Kunin. You read Wolf and Kunin. Oh, before I forget, the reason Kunin has so many publications, he has a staff of 30 scientists working for him. 30. And so he becomes...
And he has no limit of funding, right? Because he is at the NIH.
At the NIH. One of his scientists who was who probably still is an active researcher for Kunin, was my professor in bioinformatics. So it's not like I'm not acquainted with that community. And I'm just pointing that out to the viewers.
So you even have inside information.
I have inside connections.
I wanted to do a postdoc with Eugene Kunin. I applied and asked him, and he denied my application.
Was that right after you did your postdoc at Harvard?
No, it's when I was in Missouri, when I was actually an associate professor, because I wanted to learn from him. Okay, yes. Go ahead, read your abstract.
Okay, that's wonderful to know. So you're familiar with him and what his team does. This is great. And by the way, I can only say great things about my professor who worked for Kunin. My professor treated me well. I learned so much. I'm grateful for the class. So thank you very much. The abstract. Okay, so genome reduction is the dominant mode of evolution 2013. A common belief is that evolution generally proceeds toward greater complexity at both the organismal and the genomic level. Numerous examples of reductive evolution of parasites and symbionts notwithstanding. However, recent evolutionary reconstructions challenge this notion. Two notable examples are the reconstruction of the complex archaeal ancestor and the intron-rich ancestor of eukaryotes. In both cases, evolution in most of the lineages was apparently dominated by extensive loss of genes and introns, respectively. These and many other cases of reductive evolution are consistent with a general model composed of two distinct evolutionary phases: the short, explosive innovation phase that leads to an abrupt increase in genome complexity, followed by a much longer reductive phase, which encompasses either a neutral ratchet of genetic material loss or adaptive genome streamlining. Quantitatively, the evolution of genomes appears to be dominated by reduction and simplification, punctuated by episodes of complexification. Do you have any thoughts? And I'll cover some sentences there.
It's interesting, right? So especially when you read the sentence, the short, explosive innovation phase that leads to an abrupt increase in genome complexity. Well, that comment is unexpected on the basis that we know it is very difficult. Actually, no research, no experimental research, nobody has demonstrated that by accumulated mutation, one can generate a new gene, right? Nobody has ever observed a gene being created by accumulated mutation. Yes, when we compare current existing organisms, we see differences in genes, gene numbers, or different kinds of genes, and they are assumed to be due to evolution. But in reality, nobody has observed creation of a new gene through whatever mechanism, except only by comparing different organisms and assuming they evolved, right? So that is interesting when he said, well, actually, there are a lot of those processes, explosive complexification, which means that gaining of new genes, actually not only one new genes, but many, many new genes at the same time. So what we see is skimming down loss of a gene, but not gain of a gene. And then we need a lot of gains of genes to make any new things.
So like going from a prokaryote to eukaryote.
Right.
And since, well, now I will be your student. I'm going to ask you molecular biology question. So, and you've seen me present on this before. So there's import and export of, if you create a membrane-bound nucleus, then there has to be a lot of mRNAs that go out of the nucleus, that go into the cytoplasm, and then the RNA transcripts are then translated to proteins. Some of those proteins have to go right back into the nucleus. And so there's an import-export process. Those proteins need a nuclear localization signal. So there are a lot of orphan proteins or orphan genes that code for these proteins that have to make things like the spliceosome parts. But even the things that may have come from a would-be prokaryotic ancestor, it needs to have a nuclear localization signal placed on it somewhere.
Right.
So to me, this seems like that would be an abrupt, explosive innovation phase that would have to happen. And I'm just like, maybe like in one generation, because I don't know how a eukaryote could live if if you don't, I mean, eukaryote that has to implement chromatin, spliceosomes, and you can probably name all the even more parts, and then be able to even take the conserved, anything that was conserved from a eukaryote inherited, I mean a prokaryote, you have to start adding localization signals. Oh, and then plus all of your new genes also have to have localization signals. And to me, that would be an explosive innovation phase. It's an abrupt increase in genomic complexity, and I don't think we have any experiments to show that anything like that is possible. I mean, I think the reason, if we detect a eukaryote somewhere, like say in a petri dish, I don't know, do we use petri? Are there eukaryotes that are put in a petri dish? I don't know. Let's say
it is possible. Like, yes, like a cell culture, right?
well, we wouldn't presume that a prokaryote suddenly just changed into something as complex as a eukaryote. It had to be placed there. So,
well, not only that, to become a eukaryote, you need a lot of eukaryote-specific genes. Bacteria also have many. Okay, most of the bacterial genes are unique to bacteria. So you need bacteria-specific genes to make a bacteria. Not only you need the eukaryote-specific genes to make eukaryotes, because eukaryote is more complicated. Even for the simplest bacteria, they have their own unique genes. You cannot get from anywhere else. So bacteria also need their unique genes to be a bacteria.
Oh, and same for archaea.
Archaea too, yes.
Oh, so simultaneously, you have to dump all these genes that are unique to bacteria, replace them with eukaryotic versions,
right,
add some more eukaryotic genes.
Right, yes.
Anything that's carried over, you have to add nuclear localization signals that need to be localized. And then, oh my, that'd be an abrupt increase in complexity.
Right.
It's an increase in complexity, but plus losing all these prior parts.
Yes, so not only that, but also the how eukaryote organisms define whether a gene is a gene, and how bacteria define whether a gene is different. So basically, for a gene to be regarded as useful and functional in bacteria, when that same DNA sequence, if you put it into the eukaryote, according to the eukaryote criteria, those genes are nothing. They are just burdens. They are garbage.
Oh, that's right. Oh, and not to mention, like, okay, so
yeah, it's kind of like your mailing address analogy, right?
Right.
So instead of get the right label sent to the right place, maybe you get the wrong label sent to the wrong place, which is further from your destiny than closer to your destiny.
Oh, that's right, because you have, okay, I'm a little bit more familiar with the eukaryotic transcription factor. There's one transcription factor that uses zinc finger domains. And so that helps recruit proteins to the right promoter region in prokaryotes. To my knowledge, there is an analog to that in prokaryotes, but the construction is different in the way it implements the equivalent of a zinc finger domain. So the proteins that converge on the promoter region are different. Oh, not to mention in the chromatin components, that's a whole nother story. In the way you do DNA double-stranded break repair in a chromatin complex, you need chromatin remodelers. It looked like I saw at least 70 different factors needed to implement it. Let's say it's only 20.
We're getting too complicated. Maybe we can talk about those another time.
Okay, see, this really put a smile on my face. I did want to highlight one thing in Kunin's abstract. Remember I said sometimes if you want to hide something, if you want to at least clear your conscience and say something, but you don't want to get too much punishment for saying it, you use difficult wording. And he said a common belief is that evolution proceeds toward greater complexity at both the organismal and genomic level. Okay, that is a common belief. And he says numerous examples of reductive evolution of parasites and symbionts notwithstanding. So at the level of, say, elementary, junior high, and high school, you'll get kind of, when I was growing up anyway, you got the impression of evolution generally proceeds toward greater complexity. But when you start to move up into the college level, you know about evolution of parasites and symbionts. Parasites they are known to have lost a lot of genes and organs. So they have said, like the tapeworm, outside a body, a host, when it gets in the host, it starts to lose organs and digestive because it doesn't need them. Again, like the bikini, I'm going to keep using that, the bikini hiker, so people just don't forget. It's like the bikini hiker just starts dumping stuff. So that started to be well known. For a long time. But what I'm finding is now, in the era of genome sequencing being a million times cheaper, we're seeing this all over the place now, where there is gene loss. I have to, before we close, and I hope we can visit again, if you had any specific questions, and I'm sure I invite you to email me. I'd look forward to seeing my star student come back. And I'm honored to have the privilege of sharing my knowledge with you, because I've learned so much from you. And according to that passage in Romans, we're able to instruct one another. Now, some people will say, well, we have Dr. Dan Stern Cardinal. He said we have numerous examples of de novo gene emergence. And I am aware of the claims that non-coding transcripts supposedly become genes or whatever. Are you familiar at all with any of that literature?
Well, orphan genes.
Oh, yes.
When new gene, that's my area of research, right? Where gene come from, the comparisons of different organisms, gene organism-specific genes. That's my research area.
So maybe in a future show, when you have time, I might want to just talk about some of the papers Dr. Cardinal has been trying to advocate. I am also, having worked with Dr. Sanford, I had contacted Dr. Akamura, who had been trying to look at non-coding transcripts becoming proteins or whatever. And so I'm going to leave that as an open question. You said we haven't observed genes through mutation process, and I want to compare that with some of the other claims that I'm hearing. So I'm just trying to let the readers know I'm acknowledging that people have made, like Dr. Dan, have claimed that he said to me, Sal, new genes arise all the time.
Yeah, I think it's worth to really dig into details about the claims about genes. What is a gene? What is a novel gene? What has been changed? What has been observed? So details here, I think, is important.
Well, I'm going to try to spool up on that area a little bit, but if we maybe some future time, I'd be honored to learn from you, as you've taught me so many things.
Learn from each other, right?
Learn from each other, right? And you're invited to come back if you wanted to talk more about the details of fitness, and then also we can also discuss the papers that I've shown, that I've introduced here. I hope I don't know. I know you read an awful lot. I at least hope the papers that I've shared would be very interesting to you.
I will try to read them.
Okay, so that does it for this session. I'd like to thank my distinguished colleague, Dr.
Thank you so
Ching
much.
Tan, for joining me and becoming for a brief moment my star student. and glory and honor to our Lord and Savior Jesus Christ. I'd like to thank all who've joined us in this discussion. Take care. God bless you, and happy Thanksgiving
You
to Dr.
too.
Tan and all of you. Yes, take
Yeah.
care. God bless.
God bless Thanksgiving. Bye-bye.